of reactions. All the reactions are trying to achieve the higher values. That means they are trying to go for maximum values. And here also in the lowest kinetic parameter, K is less than 10, it is also going to, on an average, it is going to be around 5. Then for second one, it is going to be very close to 15 up to 200 generation. Then the third one, the maximum one, here they are trying to achieve 20. That means the rate of reaction is not going too much, but it has certain limit, and then it stays less than equal to 20 rate of reaction. Similarly, here these are the rate of the kinetic parameter distributions, which also clearly justifies the same thing as we can see on the left side figures. Now we plotted the signal versus response graph, that is also called dose response graph or curve. Here it is mostly behaving as a normal distribution. So it looks like a normal distribution. So we can say it is quite normal distribution. So it is just input we have selected, taken x-axis as the input signal, y-axis as the response, and then it behaves like a normal distribution. Next is how the kinetics of fully active proteins are changed after removal of input signal. Here what we have done, after certain time points, that means here we can see the time points are up to 500, and I think we have removed the input signal after 200 time points. Then what happens, just after the removal of input signal, in most of the networks it tries to go back to zero. That means the peak is trying to end up. So that means the activation, there is no more signal. When you remove input signal, there is no more output response at all. But there are some of the networks also which are showing the further increase in the output response, as you can see on the in generation 150 in system 1, system 2, and also in generation 50 in system 1 and system 3. So here again we are going for partially active. This is for fully active, and for partially active it is mostly showing that there is highly transient or fully adapted response. That means the peak is coming very sharp, and then it is immediately trying to go back to zero. So next part is effect of changes in the concentration levels of signaling molecules. Here what I have done is I have tried to allow the change in the concentration of input signal in each generation, concentration, change in the concentration of receptor molecules, and this one is change in the concentration of receptor molecules. And in change, when we are changing the concentration of receptor molecules, it is immediately trying to reach the maximum fitness value, that is 1.0 for the two stronger values. But when k is less than 10, then it is not exactly maximum, but it is close to zero 0.9. So what we see here, there is adaptation in output response. That means it does not always remain sustained response or always activated response, but it tries to go back to the zero values. That is called fully or partially adapted responses. Those peaks which are completely going back to zero, it is called fully adapted. Those responses peak which are partially going back is called partially adapted. So here we can see clearly in generation 1, since the parameter, kinetic parameter is so weak, so it is not showing any big change. But in generation 50, generation 200, it is clearly visible. So it can be basically seen as a change. Now it is change in the intermediate signaling molecules concentration, and then another one is change in the concentration of target molecule, that is the final molecule, that is ERK in this case. And finally, this is experimental validation for the partially activated phospho arc, like let's say single phosphorylated arc, which is like just to justify. I will not say it is accurately justifying our computational prediction, but it is somehow showing closer to our work. Next is now the dose response relationship for all the four systems. Here we can clearly see in four systems, they are behaving differently. Now, the conclusion is stronger interactions and addition of new nodes are just helping to go for improved cellular responses. Different in concentration of the signaling molecules plays crucial role in the nature or modulation of cellular responses. Now, third part is to investigate the crosstalks between the signaling pathways. So what we have done is here, we have tried to establish two pathways in parallel, and then we tried to establish the link between the two pathways, and the link could be activatory or inhibitory links. And these links are somehow showing a kind of like for better understanding. In general, the biological networks does not look like this. For example, for pathway 1, it is response 1. For pathway 2, here it is hidden response 2. So we investigated both the responses for both the pathways by allowing some connection. The basis for this connection can be seen in one of the review paper which was published earlier in the biological system, where they are showing there are four types of crosstalks, which I will be discussing in the next slide. And the method remains the same as it was in the first paper. Now, there could be different types